is the pre-lecture. As you know from my email, I am not in this week very much. So I will record the pre-lecture for this week's lab. We're going to do chromatography techniques and separation of different kinds of molecules. And so when we have products, from cell extracts or from growth media, products that have been secreted perhaps, regardless of where they come from though, we have a mixture of different kinds of molecules, and oftentimes it is something that we want to do is separate these molecules. And so you can separate them by size, and you can see here on this drawing we have the hexagons are larger than the blue pentagons, so we separate them by size. The hexagons will run out quicker than the pentagons because of their size, and that's what we'll do in lab this week. We'll do what's called size exclusion chromatography, but more on that later. We can separate by charge if hypothetically the isoelectric points of these two molecules differ. We can separate the red and the blue like that, and that is akin to what we talked a little bit about for STS-Page, separating by the charge of the different molecules. Only this applies to proteins, small molecules, whatever you want, whatever you're trying to separate. And then there are separations that can be done by other chemical properties, either how hydrophobic or how hydrophilic these two things are, or how well they bind to a certain protein, something like that. There are a variety of other chemical properties that could allow us to separate these two molecules effectively. And so we can separate the reds from the blues effectively by all these ways, providing we have information about the contents of our mixture here. And so that brings us to the whole idea of chromatography. So what are the principles that are in play here? Well, we're separating molecules. Separating one molecule from another. Depends on how many are in your mixture, but this based on some kind of chemical property. And that could include molecular size, it could include charge, solubility in a certain type of liquid or solvent, whatever. Or it could be a combination of these things. Basically what happens is the samples are going to pass over what we call a stationary phase. They're going to be carried by this mobile phase, which is the solvent that the molecules have been dissolved in. And they're passing over a stationary phase, which could be column packing material. It could be paper. It could be some kind of polymer on a sheet. The targeted molecules, though, the ones we're trying to separate from each other, are to some degree attracted to the stationary phase. So at some point the molecules will have such a high attraction that they'll just stay put, and the solvent will keep running, leaving those molecules behind. So they'll be retained relative to the other molecules, and this will happen again and again, and it depends on how many molecules we're trying to separate from each other, how many times this happens. So it's basically telling us that some molecules have more of an affinity to the solvent, the mobile phase, and some molecules have more of an affinity to the polymer, paper, whatever, the stationary phase. And so choosing appropriate stationary and mobile phases is key here. It's the essence of developing methods to be able to separate your product from everything else. Or if you have multiple products, being able to separate them from each other as well as everything else. And if we want to be general about it, there are two types of chromatography. There's preparative chromatography, which is essentially, we've been doing a lot of preparative experiments in lab. This would be separation of components for further use. So you're going to purify them further and then eventually distribute them and package them and whatever. So preparative chromatography is just getting your product ready to package and sell. And analytical chromatography is more of a fact-finding mission. Essentially, you're separating components in order to analyze what's there, to analyze that mixture. And it should be noted that these things are not mutually exclusive. You could do both. You can get a lot of data on the analytical side, while preparing your products to be polished, packaged, and whatever else. Okay, so there's some vocabulary that I'm going to use throughout this recorded lecture here. And there's probably some vocabulary you might hear around the lab, and there's some vocabulary certainly you'll see in the lab manual. And we'll start with chromatograph. Now we won't really be using a chromatograph. If you want to see a chromatograph, you can look at my lab. I have two what are called HPLCs, and those are chromatographs. These are instruments that enable the separation of compounds by chromatography. And what these do is they will pump liquid that should have some compounds in it, dissolved in it, over a column, which is the stationary phase. And this column and the liquid that's pumped over it will help separate the molecules based on the molecule's affinity to either the column or the mobile phase, that solvent. A chromatogram is a visual output of chromatography. You'll be making a chromatogram in your lab this week. Analytes, these are the substances to be separated. So every compound in a mixture that is separated by chromatography is called an analyte. Mobile phase is the solvent, that liquid phase that moves in a definitive direction, which carries these analytes. We'll be using liquid. It should be noted that we could use gas, and there's a such thing as gas chromatography as well. The stationary phase is the substance that's essentially, it says what it is. It's fixed in place. It's stationary. That allows for the chromatography procedure, allows these analytes to be separated. We will use something called Cefidex. There are plenty of other types of stationary phases that could be used, including filter paper, including cellulose and things like that. And retention time is essentially the time it takes for a particular analyte to pass through the entire system, to pass through the column. And so you'll be determining retention time in your lab this week. Retention time of molecules that are coming off this size exclusion chromatography column. We call a solvent front. This is particularly used in thin layer chromatography and paper chromatography, but it is used in other types of chromatography. It's essentially the total distance traveled by the whole solvent system. And as the solvent makes its way through the paper, the thin layer chromatography polymer or whatever, you can sort of see the solvent front progress over time. And so that brings us to talking about planar chromatography. So we'll do column chromatography this week, but planar chromatography is a separation technique in which the stationary phase is present on a plane. And again, this can be paper, this can be polymer, thin layer chromatography, that sort of thing. So again, it's either paper or layer of some kind of solid particles on a support. Whether it's glass or plastic, usually that support is inert and is not going to react or bind your analytes. And then we have what's called the retention factor if we're looking at planar chromatography. Here you can see a drawing of planar chromatography separating two analytes, and we have this gray line here is the solvent front, and that direction is the migration of solvent, so we started down here and have migrated upwards. But you can see the retention factor, and that's how far a given analyte travels. And it's as simple as measuring kind of the center of the spot of the analytes compared to the solvent front, and taking that ratio. That is your retention factor. And you can see the retention factor of the red molecule is going to be less than the retention factor of the blue molecule. So paper chromatography is a good example of this, and it's a really cheap example. You basically are using filter paper as your stationary phase. Solvent, some kind of solvent, is your mobile phase. And usually we run it from the bottom to the top, where the solvent migrates up the paper via capillary action. And so you can see we get separation of molecules here. And we can calculate the solvent front. We can calculate the retention factor of all these molecules in the paper. And typically paper chromatography is analytical as opposed to preparative. Although in some cases we could potentially do preparative paper chromatography, although it requires a lot of work here. But generally you're just trying to see how far these things migrate and separate them. Here's an example of paper chromatography. Here some spinach extract is being placed onto a filter paper strip, and in a solvent system that consists of 9:1 ether and acetone, we are migrating up the paper, and we see the separation of a bunch of different chlorophyll compounds. And you can see that here in this picture, and you can see this here a little more sloppily, however. You can see that here in this picture, that the chlorophyll compounds are separating based on this solvent system. Thin layer chromatography is similar. The stationary phase is different. It's made up of something else. Often it's nitrocellulose or silica or some other polymer that's attached to an inert substrate. Often that substrate is glass. And again, like paper chromatography, thin layer chromatography is usually analytical. And you can see, just like with paper chromatography, we are placing our compounds or our mixtures down at the bottom, and we are allowing the solvent to migrate up the thin layer via capillary action over time, and that separates the compounds, and then we can calculate our retention factor, again based on the solvent front. And you can see how that's done here. Here's an example of thin layer chromatography in the literature. So here we're looking at lipids and fatty acids that are present in palm oil that has been used by bacteria for a growth substrate. And we're seeing how quickly we get rid of, or how quickly the bacteria break down triacylglycerols. And here we're separating the triacylglycerols, or the fats, from the diacylglycerols, from the monoacylglycerols, and from the free fatty acids. And we're using two different types of solvent systems here to move the analytes up the thin layer chromatography sheet. One is chloroform, methanol, and water, and we're going 5 centimeters with that. And then the rest of the way we go using this solvent system: hexane, diethyl ether, and acetic acid. And you can see what kind of separation that gives you. And so how are multiple compounds separated? Well, these compounds can be separated based on the competition of solute and mobile phase for binding places on the stationary phase. So the mobile phase is attracted to the stationary phase, and the analyte is attracted to the stationary phase. If you are looking at polar interactions, you would use a polar stationary phase like silica gel. If you're looking for a nonpolar interaction, you use nonpolar stationary phase. For example, this polymer C18 is useful. Okay, so we have polar compounds and polar stationary phases here. If we are separating polar analytes, we use silica gel. And here you can see a portion of silica gel as it would be on the thin layer chromatography. And you can see it is rather polar. It has some hydrogens and oxygens. So it can form some hydrogen bonds with your analyte. And so what you would separate, you have the more polar component would get hung up because it is very attracted to the polar silica gel. The less polar component ends up migrating further because the solvent can just bring it along. So a polar analyte compound wants to bind to that polar silica gel. And in TLC, it will not migrate up as far from on the stationary phase. Usually a mixture of solvents is preferred. We don't just use one solvent. And typically that choice of solvent in thin layer chromatography is trial and error. Or you look up a paper of someone who has done a similar separation long ago and use their solvent system and see how that works. And so you would typically use some mixture of polar and non-polar compounds. Like before I talked about using water as one of the components, as well as some more hydrophobic things. This is typically what's done. We have a mixture of different types of compounds as TLC solvents. And these lead to pretty good separation of compounds. You can see why that is. So here we have a bunch of thin layer chromatographies here, and we're using different solvents. And so it's a polar stationary phase. And so if we're using 100% hexane, which is entirely hydrophobic, if it's too nonpolar, you won't get much movement of your solutes. You'll get a little bit, you don't get good separation, as you can see here, because the solutes or the analytes would rather bind to the stationary phase than they would to the solvent. And the solvent's, of course, not interested in binding to the stationary phase, so it leaves the solutes behind. And if you're too polar, we have 100% ethyl acetate here, then these analytes move far too much, let's say, and you can't get good separation of your mixtures. You don't get a good retention time or retention factor. You don't want to do too polar either. A better thing to do is a mixture of the two, and you can see what a mixture of the two gets you: very good separation, where you can see the reactant, the product. And if you're looking at products of a reaction mixture, you can see, here's the reactant, here's the product. We can see reactant being turned into product. Good separation. We're using one-to-one mixture of this non-polar and this more polar solvent here. And so we can determine this looking at eluent strength. And you don't always want the largest eluent strength. This is for the polar stationary phase silica gel. Of course, hexane is the strongest, and you can see what that gets you on the previous slide. Pentane's a little bit less strong, but it's still pretty strong. Carbon tetrachloride, benzene, diethyl ether, acetonitrile, then acetone, butanol, methanol, and finally acetic acid. Now, if we're using a hydrophobic plate, C18 let's say, then the order of solvent strength would be reversed. Now again, you don't necessarily want to use the strongest eluent here when you're doing separations, but maybe you want to have a little bit of it in there in the mixture. It depends on what you're trying to separate and how much you're trying to separate them. And again, I said this is all mainly trial and error here in terms of TLC and picking a good solvent. Like with, I mentioned 2D gel electrophoresis, we can do 2D thin layer chromatography, where we separate based on one principle in one direction and then separate based on another principle in a second direction. And we can get good separation of a variety of different mixtures, a variety of different analytes and a variety of different mixtures. Okay, and so now let's talk a little bit about chromatography techniques that are determined by the physical state of their mobile phase. And there are two main ones that I want to go over. There's gas chromatography and liquid chromatography. Pretty much everything falls into one of these two categories. Even things like thin layer chromatography can be considered liquid chromatography in a way, because your mobile phase is a liquid. And so here are the setups of what these two chromatographies look like. And again, you can see you have a gas carrying the analytes in gas chromatography. You have some kind of solvent or liquid carrying the analytes in liquid chromatography. So first let's talk a little bit about gas chromatography. This is analytical chromatography, mainly because it's going to destroy your compounds. And separating and analyzing compounds that can be vaporized without decomposition is the goal of gas chromatography here. So we can vaporize them and see what they are, and that's the main objective for gas chromatography. The mobile phase is typically some kind of relatively inert gas. Helium is often used, although most gas chromatography machines I've run use hydrogen as the mobile phase. The rate of progression throughout the gas chromatography column depends on the strength of adsorption, the adsorption to the column walls, or the packing material in a column. depending on what you're using. How do you detect the compounds? We can detect them using this thing called the flame ionization detector. That's the most common. And so this detects hydrocarbon-containing compounds. So it's good if you're looking at organic compounds. For example, if we're looking at an analysis of gasoline, what's in gasoline using gas chromatography, you can see you have a variety of different alkanes. Hexane, heptane, octane, and some nonane, and maybe this is decane out here, I don't know. But you can run them through the gas chromatography. They'll stick in varying areas to the GC column wall, and that will help separate them, these compounds, from the gasoline mixture. So when they reach the flame ionization detector, we can detect individual compounds. Liquid chromatography. High performance liquid chromatography is usually what you hear about, otherwise known as HPLC. This can separate, identify, and quantify components of a mixture, and we're looking in the liquid phase. It's again based on principles of adsorption to some extent. Pumps will pass pressurized liquid through a column that's filled with some sort of sorbent, and that sorbent is the stationary phase. And that sorbent can attract either charged molecules, or it can attract hydrophobic molecules. Again, it depends on what sort of separation you're trying to do. There are a variety of detectors you can use. There's UV visible, and we have looked at the compounds in UV vis spectrophotometry, and I've shown you some peak wavelengths in which you can look at given compounds using the spectrophotometer. The principle is the same as a detector in liquid chromatography. It's just like a spectrophotometer, UV vis spectrophotometer. Or you can use what's called a photodiode array to detect molecules coming off the HPLC column. HPLC can be highly preparative, and there are a variety of ways we can do this. We can look at a mixture and run it through a column. Either we can run it through 3 columns in series, and you can see with every cycle we're getting better separation until the point where we've separated the 2 molecules. Or we can run the mixture 3 times through the same 1 column, and again achieve this separation here if we want to do that. And so we get better separation of components. We can use multiple columns, it's more expensive, or we can just recycle and use the same column 3 times or more. But then we have our separated compounds. We can then further prepare them as products. Here is an example. We're looking at HPLC detection of depolymerization products of a biopolymer called polyhydroxybutyrate. This is enzymatic depolymerization done by bacteria using a natural enzyme called FAZ1 and some other proteins that do this. So we're looking at the mobile phase, which is a mixture of ammonium acetate and methanol. And what we see here is we get some CoA in the mixture. We get this 3-HB-CoA, which is the monomer of polyhydroxybutyrate. And we also get this crotonil-CoA, suggesting that when PHB is depolymerized, it usually comes off in the form of crotonil-CoA. Ion exchange chromatography is very frequently done, and that separates proteins that have differences in charge, or it separates other molecules that have differences in charge, although it's pretty effective with proteins because there's a wide variety of isoelectric points here. And so this is based on a reversible interaction between protein and the chromatographic medium. These are all based on some kind of molecular interactions. This is why it's important to understand what molecular interactions are in play here. And you can see in the picture that it looks like what they are doing is basically a gravity flow ion exchange chromatography. So we're going to do gravity flow chromatography, but it's not going to be ion exchange, it's going to be size exclusion. And we'll talk about that in a little bit. And so in ion exchange, proteins get bound as they load are loaded onto the column. They get bound to these charged polymer beads, these charged particles. And then we alter conditions by changing the salt concentration, changing the pH of the buffer we're using, and that allows substances or our analytes to elute differentially based on their charge, whether they have a large net positive charge, a net negative charge, net positive charge, or a large net negative charge, depending on what they're doing. And they will come out in different fractions here. And changes of conditions can be made either stepwise, or if you happen to have a gradient maker, you can do this in the form of a gradient. And here's what this looks like if we're looking at a column that has the stationary phase on the walls. We have a mixture of, say, amino acids, which will all have some kind of different charge. We have cations immobilized on the surface of the wall here. Solution flows through. You will bind the negatively charged ones right away, while the positively charged ones kind of go through. And then that separates the negatives from the positives here. Alright, let's see if I can do this. I have an ion exchange chromatography video I can try to show here. I've never tried to do this in a recorded lecture. We'll see if I can get this up and running. It looks like I might be able to do it. So here goes.
Separation in ion exchange chromatography depends upon the reversible adsorption of charged solute molecules to immobilized groups of opposite charge. Most ion exchange experiments are performed in 4 main stages: equilibration, sample application and wash, elution, and regeneration. The first step is the equilibration of the stationary phase to the desired start conditions. When equilibrium is reached, all stationary phase charged groups are associated with exchangeable counterions, such as chloride or sodium. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. The sample buffer should have the same pH and ionic strength as the starting buffer, in order to bind all appropriately charged proteins. In the third step, elution, biomolecules are released from the ion exchanger by a change in the buffer composition. A common way is to increase the ionic strength with sodium chloride or another simple salt in order to desorb the bound proteins. Proteins are desorbed relative to the number of charged groups on their surface. The final step, regeneration, removes all molecules still bound. This ensures that the full capacity of the stationary phase is available for the next run. Separation in ion exchange.
Okay. I don't know how well you heard that video. This is the first time I tried to do this in a recorded lecture, so I will try to post these videos on my courses if you want to take a look at them and you need better sound. If you don't need better sound, don't worry about it. I'll still try to post them anyway. But what we're going to do today is size exclusion chromatography, or SEC. And that separates compounds by size or molecular weight, typically. It can also be called gel filtration chromatography, or it can be called gel permeation chromatography. And essentially, we let a mixture of molecules, some small, some large, we let it loose on a column that have these porous polymer beads in it. And the smaller molecules, proteins or whatever, are going to get hung up in these pores. And so they're going to take the long and tortuous path through the size exclusion chromatography column. Whereas bigger molecules, larger proteins, aren't going to be able to get into those pores. So they are going to kind of take the easy way through the size exclusion chromatography column. And that allows the large guys to come out first and the small guys to come out last. And you can see that here, we dump a bunch of particles onto some polymer beads, some gel beads, and they begin to separate as we flow through the mobile phase. The large guys are going faster because they're taking the easy route, and the large molecules end up eluting off the column first. And hopefully that's what you're going to see when you do the size exclusion chromatography in lab this week. The small molecules are getting hung up in the pores in the matrix, and they're taking the long way in. And so they will come out, they'll elute off the column at a later time. And you can easily separate the two types of molecules. And let's look at, again, I hope the sound is sufficient. If it's not, I plan on posting these online. So here, let's look at the size exclusion chromatography video right here.
Separation in gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed with a chromatographic medium, which is a gel. Molecules larger than the pores in the gel are unable to diffuse into the gel and are confined to the solution surrounding the beads. Molecules which range in size between the very big, excluded from the gel, and the very small can penetrate the pores to varying degrees based on their size. Access to the pores is limited by steric hindrance. If a molecule is smaller than the smallest of the pores in the gel, it will be able to enter the total pore volume.
Okay, and so we can separate proteins, but what if we want to know what molecular weight our protein is? And generally we can do this by using what we call standards. So proteins are some kind of molecule of a known molecular weight. And generally this is how size exclusion chromatography columns are calibrated, using 4 to 5 standard samples. These are folded proteins often of known molecular weight. In some cases you can get styrofoam particles that are of known molecular weight. But regardless, the idea is the same. You're calibrating using molecular weight standards. So when your protein comes off the column at a given time, you can estimate its molecular weight, if you don't know it already. So a sample with a very large molecular weight protein would be used to determine what we call the void volume. So basically that very large molecular weight protein, like I said, is going to take the easy way through. It's just going to go through what we call the void, or that area in the column that isn't taken up by the stationary phase. So the large protein is going to go through that, and that will determine how long it takes to go through. And then that can also help us determine what time it takes to get through the void. The elution volume of proteins will decrease in a roughly linear fashion with a log 10 of molecular weight. And this is due to the molecular hydrodynamic volume here. So what does this look like? Well, so void volume, strictly speaking, is the volume of space between the packing material inside the column, in the column walls, and things like that. So again, we inject a sample. We are separating by size. When large solutes start to elute, that is the void volume, because they have basically gone through the void. And the elution volume would be the volume, and this void volume is the volume of buffer that has passed through before the largest sample has come out. That is what the void volume essentially is. The elution volume is going to vary depending on the sizes of your smaller solutes. And the elution volume is the volume of buffer required to pass through the column before whatever molecule of choice elutes. And the more molecules you have eluting, the more different elution volumes you're going to have. A quick aside on hydrophobic interaction chromatography before we get into what we're going to do in lab. This separates proteins with differences in hydrophobicity. This is based on a reversible interaction between protein and hydrophobic surface of chromatographic media. The interaction is enhanced by the ionic strength of the buffer. So a high ionic strength buffer keeps those hydrophobic interactions nice and tight. We then alter our conditions. We change the buffer to elute what's been bound. And this is usually performed by a decrease in salt concentration that allows the hydrophobic molecules to pop off the hydrophobic interaction chromatography column. And the principle of the thing is we are absorbing onto this hydrophobic interaction chromatography media, and you can see there's a hydrophobic stretch of perhaps hydrocarbon here that is interacting with a hydrophobic protein. And then we can desorb via an ionic repulsion, so this can bind a metal ion or combine some sort of ion that will then pull off the hydrophobic protein that we're trying to purify. And so if we have the ionic strength is up, we essentially have the hydrophobic interactions here as they adsorb to the different types of hydrophobic interaction molecules. We decrease the ionic strength, we end up with desorption because that is changing the charge of the thing that we're trying to purify, the protein we're trying to purify. So it repulses from the hydrophobic interaction column, and we can get it off the hydrophobic interaction column. Alright, so I'll try one more video here. Let's see if we can do this. And again, we'll try to put these online.
Separation in hydrophobic interaction chromatography depends upon the reversible adsorption of biomolecules according to their hydrophobicity. Most hydrophobic interaction chromatography experiments are performed in four main stages: equilibration, sample application and wash, elution, and regeneration. The first step is the equilibration of the stationary phase to the desired start conditions. Equilibration is performed by adding salt to the mobile phase. The general equilibration buffer in this step is 50 millimolar sodium phosphate with 1 to 1.5 molar ammonium sulfate. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. The binding is promoted by moderately high concentrations of salt. A concentration of salt that gives an optimal purification is chosen. In the third step, elution, biomolecules are released from the hydrophobic surface by a change in the buffer composition. A common way is to decrease the salt concentration of the buffer. Gradient elution can be used to separate the target protein from other bound proteins. Normally, a gradient of 10 to 15 column volumes is sufficient. The final step, regeneration, removes all molecules still bound. This ensures that the full capacity of the stationary phase is available for the next run.
Okay, one more chromatography to get to here. Affinity chromatography, and some of you will be doing affinity chromatography in the end of the semester project, the group project that we will be doing. There are a variety of types of affinity chromatography that can be done. We'll do one that involves binding to nickel binding. But anyway, the idea behind affinity chromatography is that we're purifying compounds or proteins, usually, based on their affinity to a matrix-bound substance. The matrix in the column will bind that protein or compound fairly specifically, and then we can elute that protein or compound by some way. And with nickel, you're changing the ionic strength and you're changing the buffer composition, and that's helping you do that. And so essentially what you're doing is that we have a matrix with which something called a ligand is immobilized onto, and you have a protein, or your sample will attach to that ligand and will form a complex. So this will stay behind, your sample, the thing you care about. All the impurities will flow through and get washed out. And then when you're ready, when you're satisfied that the sample's been purified well enough, you can then add a new kind of buffer to release the sample from the ligand. You can alter the ionic strength. You can put in something that will compete for binding with the sample or the ligand. Depends. And here's what this might look like here. We have a sample mixture that's put on a chromatography column, and again, depending on what comes through, you have different types and volumes of effluent and eluent. We wash out all the impurities, the stuff we don't care about, and then if we change the buffer to some high salt elution buffer, we can then elute different things that have been attached to our ligand that was immobilized in the column here. And so if something doesn't bind the ligand very strongly, it'll come out first, but things that will bind the ligand strongly and need a lot of that high salt elution buffer will come out last. And usually these are the most important things that you want. And you'll see how this works if you're one of those that is doing affinity chromatography in the end of the semester project. And so this week's lab is the separation of molecules by gel filtration chromatography, or size exclusion chromatography. The stationary phase for this is something called Cephidex G75 beads, and these are those porous polymer beads that are going to hang up your smaller molecules and make them take a tortuous path down the column. Your mobile phase will just be a simple phosphate buffer with no need for anything fancy here. We're going to run these columns under gravity flow, and you've seen those burettes in the lab that are attached to a stand. We're going to use those as our chromatography column. So you're going to pack your chromatography column first thing, then you're going to add some buffer. You're going to run the buffer plus the mixture of molecules, I should say, not necessarily proteins. Blue dextran is one of the molecules. Cytochrome C is another one of the molecules, and the potassium chromate is another one of the molecules. And we will give you the absorbance peaks so you can find each of the analytes, although it should be somewhat obvious because these compounds are pretty well colored. But we'll give you the absorbance peaks so you can figure out where the largest peak is in your fraction, where it comes off. And so the principle again is you've got very small molecules are going to enter many pores of the gel and get hung up and take longer to go through the size exclusion chromatography column. Medium-sized molecules will enter some of the bigger pores, but they won't get hung up as much as the very small molecules. Larger molecules are basically going to be used to determine the void volume. They can't really enter pores, so they're going to travel rapidly through the column and will enter the bottom of the column here and elute sooner. And so here is Cephidex. We'll have a bottle like this that has this Cephidex in it. It is an inert gel filtration chromatography medium. Its upper limit of what it'll separate is 80,000 Daltons. So that's fairly big in terms of proteins. And I don't remember. I think maybe that's what the blue dextran is around, 80,000 or maybe even a little more. But anyway, essentially you'll set up a gravity flow column like this, where you pack the Cephidex column and you run the colored compound through. You won't be collecting in a flask, you'll be collecting in a series of tubes. There'll be different fractions of 1 milliliter, 2 milliliters, what have you. And then you'll put together a chart like this, where you look at the different fractions and you take absorbances at certain wavelengths, and that'll tell you whether you have blue dextran, it'll tell you whether you have cytochrome C, or whether you have the potassium chromate. And so coordination with this is key here. You need someone to be timing, you need someone to be looking after the volume, and you need someone to be running and doing the absorbances. So it's going to take a concerted team effort to get this lab accomplished properly, and so good luck with that. And so what we've talked about here is the separation of molecules, mainly using affinity to a mobile phase or a solvent, and affinity to a stationary phase. And the analyte, or the stuff we're trying to separate, they are attracted somewhat to both. And it all depends on how much everything is attracted to everything else, where you separate the analytes. Planar chromatography, which we won't do, involves things like paper chromatography, thin layer chromatography, which we talked about. And you can calculate retention factor, which shows you how far these things have gone. Column chromatography, which is what we will do. We can separate by size, and that's what we're doing in lab. But you can also separate by charge of a molecule, how hydrophobic it is, whether it binds to certain molecules. So there's a variety of things you can do with chromatography. Okay, so that is it. And good luck. I will not be in Tuesday's lab. I hope to be in Thursday's lab, so hopefully I'll see everyone running some good chromatographies then. If you have any questions, feel free to email me or ask Jayashri, and I will see you on Thursday.